Hi all! In this video I will be explaining the effect that a frameshift mutation will have on the protein sequence of a cell. First, here are some definitions for context. A frameshift mutation is the insertion or deletion of one or more base pairs in a gene, shifting the reading frame for all codons following the mutation site. A codon is a series of 3 nucleotides which together code for an amino acids, and amino acids together form proteins. Starting with the amino acid sequences for the normal protein and the mutant protein, one can decipher the specific nucleotide sequence of the amino acids using a codon table. There are many methods to do so, but this is the one that works for me. First, I start by writing out the complete nucleotide sequence from the amino acids given using the codon table. For certain amino acids, there are more than one possible third codon, so I list them beneath it as shown. Next, I do the same for the mutant protein right underneath the normal protein, just so it is easier for me to trace where the mutation is. In this drawing, the blue nucleotides are the ones before the mutation, and the red nucleotides are the ones after the mutation occurs. Next, in order to identify if it is an insertion or deletion of a nucleotide, I trace the movement of each nucleotide after the mutation. This uracil has to correspond to this uracil, and so on and so forth. Now this is when we start getting some answers. Since the next nucleotide in the mutant sequence has to be a cytosine, then we know that the original normal sequence was a cytosine instead of a uracil. I then continue to trace the nucleotides together until I decipher the entire sequence. For the stop codon, we know that the second and third nucleotides are adenines, so using the codon table, I see that there is only one stop codon that has those options. This means that uracil is the first nucleotide for this stop codon. This also tells me that uracil is the third nucleotide for the threonine in the normal protein. In conclusion, we can see that this mutation was caused by the insertion of an adenine nucleotide right after the tryptophan amino acid.